As Raphael mentioned, I'm at this place here, which actually is now called the Wolfson Center for Mathematical Biology, and we work on a number of topics, but what I want to talk about today is modeling collective cell motion in biology, and I'm going to talk about 4 examples, so you can either view these as being 4 biological problems that we try to model to solve or to investigate, or you can view them as 4 different frameworks that typically people use from a modeling point of view to address problems in biology, and I'll explain what these things are as we go through. So we'll start with the first one, which is acid-mediated invasion, and this is something that is quite an old idea now, so some of you have already heard me speak about this, but I want to mention some new results. So this was an idea that was put forward by these 2 people who are physicists, Gatenby and Galinsky, but Gatenby is actually a radiologist, so he runs a cancer unit in the Moffitt Cancer Center. And they were looking at the interaction within the body of tumor cells and normal cells. And they thought about it in a very abstract way, and they asked the question, well, how could you, if you are an invading species, so if you think of the tumor as being an invading species, how can you overcome the resident species, which is the normal cells? And there are only 2 ways you can do it. You can either proliferate faster than the other ones, or you can kill the other ones. And the problem is, from the tumor's point of view, is that the body is set up to support the normal cells. It's not an environment that supports the tumor cells. So he asked the question was, why is it that in many cases the tumor cells have what is called, generate energy via this pathway, the glycolytic pathway, and one of the byproducts of the glycolytic pathway is lactic acid. So the idea was that he put forward was that the lactic acid might actually change the environment and make the environment supportive of the tumor cells and not of the normal cells. So basically the idea here was the tumor cells go into an environment which is hostile to the tumor cells, and the tumor cells change the environment so that it becomes good for them and bad for the normal cells. So they wrote down an equation, a system of equations here. So they model the, everything is modeled as a continuum. So you've got normal cells N1, tumor cells N2, lactic acid L, and everything else here is a positive constant. So you've got the normal cells, which have logistic growth, and they produce lactic acid through this term here. The lactic acid diffuses. This is in 1 dimension, but obviously you can extend it to n dimensions. And the lactic acid in their model kills the normal cells, and that creates a space for the tumor cells to move. And normal cells don't move, which is the normal, that's their property, they don't move. So then when they analyzed this, so they started off with, I should have put initial conditions in here, sorry, I forgot. The initial conditions were that you had normal cells everywhere, so these are the normal cells, and then you introduce some tumor cells. And after a while, they found they got 2 different types of behavior, both of which were traveling waves. So here you have the tumor cells form a front, and that's traveling. Leaving behind it, the normal cells have now decreased from 1 here to 0.5. And then here's the acid. And then the other behavior they have is this one here, which is a competitive exclusion. The tumor cells have totally annihilated the normal cells, move along, and you see here a prediction that there should be a gap between the tumor cells and the normal cells. So that's a prediction, two predictions of this model. One is that by creating this lactic acid, the tumor cell is able to invade. And the second prediction is that in some cases, you get this gap. So then they went and did experiments, and they found that roughly speaking, in 1/3 of the cases, they see a gap. So here's the gap here. Okay? Now, this is a bit controversial because some people will say, some people do say, that the process that was used to collect this figure, which involved putting chemicals into the tissue, this is called fixing the tissue, that this was an artifact of the fixing. Okay, so it's an experimental artifact, people will say. But they addressed this problem in their paper by using this picture here, where you can see that it's not so clear now that there's a gap, but this is done without fixing. It's done a different way, okay? So the gap here is a lot smaller. It's not as spectacular as this. So a natural question to ask is, can we find out whether this sort of gap is possible in the model, or is this sort of gap more possible? Or is this an artifact? Okay. In fact, here are some pH values. So this is, the tumor is here, the normals are here, and this is pH taken at cross sections, and pH being like this means the acid is like that, which again qualitatively agrees with the result. Okay. And in fact, very recently, this was just in March of last year, they continue to work on this. And if we look at this picture here, we see this is tumor growth. And you can see that where tumor growth is high, pH value is low, so it's very acidic. Where tumor growth is low, tumor value, or pH value is high, so it's less acidic. So again, there's a link between, seems to be a link between the growth and the acidity. Okay? And then an obvious corollary of this is that if acidity is being used by the tumors to invade, then if you add bicarbonate, you should neutralize the acid and slow down the invasion. And so here, probably look at this picture here. What they did then was this is work by Ian Roby and colleagues, is that they put tumor cells into a mouse, implanted the tumor cells in the mouse, made them fluorescent, and then looked at metastases, looked at how these cells invaded. And what you find here, these are mice that were not treated with bicarbonate, and you see this is the number of invasions, number of metastases, and these are mice that were treated with bicarbonate, and they were treated by constantly, every time they drank water, the water had bicarbonate in it. And you can see there's actually statistically significant reduction in the invasion, but there still is some invasion, and of course we would expect that because we would expect that this is a complicated process, more than one player, more than one process is happening here. So this would suggest that perhaps the acid-mediated invasion is playing a role in this system. Okay. So what we did was we took a actually, there has been some mathematical work done on this, on looking at the traveling waves, and we developed a more realistic model because one of the problems with the original model is it always gave invasion. So you could never have a situation where the tumor died out. And they recognize that. That's very easy to fix by putting these competition terms in. We also made the model a little bit more realistic by putting in the fact that the tumor cells themselves are not fully resistant to the acid, so the tumor cells will die at some point if there's lots of acid, and that the normal cells will have some resistance, so this delta 1 should be 0 for small values of W, and then becomes non-zero as W increases. Okay? So now if you take this system and try to analyze it and look for traveling wave solutions, and what we want to try and find with this system is by putting, if we take the gap that they observed in the fixed model, in the fixed picture, and in the non-fixed picture, and if we ask the question, what parameter values will give you those two answers? And then are those parameter values realistic? Okay?
So what's the W again?
So W here is the lactic acid. So sorry, I changed from L to W. So W here is the lactic acid. And so these are the same equations as before, except there's a competition term here. So U and V are competing against each other. And the V, which are now the... Sorry, I should explain. So this now, U is now the normal cells. So U is N1 and V is N2. So this is the tumor cells, and this is the acid. We make the assumption that they too can die from the acid, but at a much slower rate than the normal cells can die. Okay? So what we find here is we get these extra behaviors. Here's a behavior where the tumor gets cleared. So if we ran this simulation, this would go to 0, and the normal cells would go back to being 1. So 1, the healthy tissue is a stable, steady state of this system. Here's a case where the tumor just stays at 1 point, so it's like a benign system. And then here's the systems, again, that they got. Okay? So these are the two extra behaviors we get. And,
There's no gap.
pardon?
There's no gap.
No, well, in this one there's no gap, but they are parameter values in which there are gaps. Okay? So I don't want to go into the detail. What you can do is you can do an asymptotic analysis of the system, and it's quite a complicated analysis. I don't want to go through the system, because if I do, you'll lose the will to live. If I go through all the details, I mean, I know for a fact I will. And what you can then do, the details of these diagrams aren't important, but what we can do then is we can analytically determine for parameter values in parameter space, how does the tumors invade? Where do they invade? What is the speed of invasion? And what is, if there's a gap, what is the distance of the gap? So we can determine all of those in terms of all the parameters, because we can do the analytic study of the system. Okay? And we can get pictures like this. So this is, for example, the gap width. How does the width depend on different parameter values? And here's the value where we get realistic gaps. These are the gaps that are observed experimentally. Okay? And the conclusion of all of this, so I'm not going to go into all the detail, I'm just giving you the conclusion. The conclusion is that to get the wide gap that they observed experimentally, you would have to have such low acid concentrations that physiologically you wouldn't expect that. Okay? So that does suggest that the wide gap is an artifact. But the thinner gap you can get for realistic parameter values. Okay? So it is suggesting that really it's the thin gap that's realistic, not the thick gap. Okay? Now there's a problem with all of this, and it's a problem that me and everybody else brushes under the carpet. The gap is only 2 or 3 cells thick, 2 or 3 cells wide. Okay? And we're using a continuum approach here. So a continuum approach is valid over length scale of 10 or 100 cells. So in that framework, you wouldn't be able to see a gap of 2 or 3 cells. So does it really make sense to say that I've got a continuum model that predicts a gap of the length of 2 or 3 cells? The answer is no, it doesn't make sense. So a few weeks ago, I was teaching this to our doctoral training center students, and then they have to do a project in a few days. And I mentioned this problem with this particular thing, and I said what really somebody needs to do is to do an individual cell-based model where you put all of these details in, but you have individual cells, and then you ask the question of, do you see the gap? And I assumed, as we always assume, that the students didn't bother listening to that. It was at the end of a lecture, end of a long day, last 2 or 3 minutes when students are all asleep or playing on their iPads. And then they start to do their projects, and on the second day of the project finished in a certain day, and as I was leaving the room, one of the students went running up to me, physically grabbed me, and said, "Don't leave, come," pulled me. And what they'd done was, which really shocked me, they'd actually listened to what I'd said, and they'd done the individual-based model, and they'd showed that there was a gap, and they'd done the statistics of the individual-based model, and the statistics exactly matched the continuum model. So I was quite shocked, and in fact I actually walked back from the Rex Richards building to St. John's with a big grin on my face. And I'm sure people who were walking past me probably thought, this is a very strange man. We better stay clear of him. He's got this funny smile on his face. So now the question is, okay, so that's fine for the mouse. What about the human? I don't want to go into details of this. It's just to say that it's bad news, not such good news. We did a model of this for the human, and 2 things came out of it. One is, you just have to look at this picture here. This is the change in pH, and this is the mouse tissue, the mouse tumor, I should say, when you apply the bicarbonate, and you see there's a big change. And here's the human. So the prediction in the human is that there's very small change, which means that the model is predicting that this will not work so well in the human as it does in the mouse. And then we said to our experimental colleagues, well, that's okay, that's no problem. It goes back to what Raphael was saying earlier on about the fish. We said, well, why don't you just double the dose? And useful to talk to biologists who then pointed out this would actually kill the patient because we didn't know that bicarbonate is poisonous. Okay? But another result, so this result still needs to be verified, because obviously you can't really do experiments in humans. But another result that came out of this model was this is the mouse blood. So this is the system of the mouse. And it's very high, the pH change here. And that's dangerous to the mouse. And then that was validated in a paper with our colleagues in Roby, who's an experimentalist, and 2 experimentalists, Bob Gillies and Bob Gatenby. Okay. So what we can do with this model is we can then think about combination therapies. So think about different, not only adding the bicarbonate, but bicarbonate is going to be poisonous if it's added in high densities, then high concentrations, then don't add so much bicarbonate, but maybe add other inhibitors. And what we did then with this model is we've come out with a number of predictions. These haven't been tested. We've been trying to get funding to test for this, and in the latest round of funding that our experimental colleagues put in, it was rejected because one biologist said, well, everybody knows the acid-mediated invasion hypothesis. It's well established, so what's the point of doing more experiments? And the other referee said, there's no evidence to show that acid-mediated wave hypothesis exists, and therefore this shouldn't be funded. Okay? So both were funded because it was right and because it was wrong. Okay? So only in biology can you be right and wrong at the same time. Okay? Which is really good, because then you're always right and always wrong, but you just think about the fact that you're always right, whereas in mathematics mostly you're wrong. Sometimes you're right. Okay? So now I'm going to go on to talk about neural crest cell migration, and there's a link between this and the tumor stuff. because neural crest cells, this is work actually, I should say, done in collaboration with experimentalists at the Stars Institute. Neural crest cells are the precursors to lots of tumors, lots of very aggressive tumors. And what happens in very early development is cells emerge from the neural crest along here, and they go to different parts of the body. Okay? And they travel long distances. And importantly, they travel long distances, and the travel is controlled. And the question is, we don't know how it's controlled. And typically in biology, it's not one system working all along here. As you move along here, there are different processes going on and different properties of the cells as they move. So we're looking at the cranial neural crest. These are ones that originate near the head. Okay? And so their properties and how they move are different to the properties, for example, of the trunk neural crest. Okay? And this diagram here is just to show you that Paul can generate lots of experimental data. So he can generate data on how the domain grows as this is happening, because obviously the embryo is growing. He can generate data on how the cells move, how different cells move, what their orientation is, etc. So we have all these data. And so originally, this is how it was thought that the process happened, how the process was controlled. It was thought to be like this. The idea was that these cells emerge, and they emerge under something called the ectoderm, which is a tissue, and this tissue produces VEGF, which is a chemoattractant. And what that means is that cells move up gradients of the chemoattractant. So the idea was the following. The cells would come out of the neural tube, they start to eat the chemoattractant, and that creates a gradient, and then they continue eating, and they move up. Okay? So if you think about there being some very nice food, and you come out and you start eating the food, then you're going in this direction because that's where the food is. So we thought, well, okay, let's see if that works. So what you're going to see here is this is the domain, and the domain is going to grow based on the biology, the details we know from the biology. This is going to be the concentration of the VEGF, and cells are going to come out of here, and cells are going to have these look like swords in front of them, and that's to represent Philopodia. So the cells are going to sample around, and where the chemical concentration is high, they're going to move up that gradient. And they're also going to eat the chemoattractant. So the chemoattractant is going to be solved by a partial differential equation, which describes diffusion and the degradation due to the cells eating the chemoattractant. Okay, so now let's see if this video works. Okay, so these swordy people are coming out here. So that looks very good. You see the cells are coming out. They're invading the tissue is growing. And you see here the amount of VEGF is decreasing. So it looks very, very good now. Everything's happening. But now you begin to notice something's not quite right. These cells are getting left behind. They're getting left behind because there isn't enough chemoattractant here. So there's no signal for them to follow. Okay, so if you look back here, you see that this model predicts actually there's going to be cells stuck here. So then we came up with a new model where we said that cells at the front respond to the gradient of chemoattractant, but cells at the back, they just grab hold of the cells at the front and follow along. And now let's see if that works. So it'll start the same as before. So these are yellow cells. These are the leaders. And that's happening as before. And now these white cells, these are the followers. Okay? So you see the followers come in. And now you see you get the invasion. So the model prediction here is that the cells at the front are different from the cells at the back. So this motivated our colleagues to do experimental analysis of the cells at the front.
And they go to the center just because of the motion of these swords or something.
Sorry, they
actually go faster in the center.
Yeah, so that's actually a good point. There is a, go back to here, there's some sort of inhibitor here and here, which people don't know what it is. That means that the cells can't go near here, so they're pushed in towards the middle. And there's still an issue as to exactly what that inhibitor is. Yeah, so somehow there's some sort of inhibitor that makes them go along, almost like along the middle. Yeah. Okay, so this is just to tell you that they did a lot of experimental work, and what they found, they analyzed 84 genes, and they found that the expression of 19 of them were significantly different in the front compared to the back. And crucially, one of these was one of the cadherins. And one cadherin that was upregulated in the front cells is a cadherin associated with cells moving in a sort of discovery phase. And one of the cadherins, and that was downregulated in the back cells, but in the back cells, a different cadherin was upregulated, which is to do with stickiness. And that was downregulated in the front. So in other words, it was exactly how the model predicted the behavior at the front. The genes regulated at the front are ones that make the cells behave as the model predicted. The genes regulated at the back are the genes that give the sort of behavior that the model predicted. Okay, so then we decided, well, now let us define some experiments. So we had a meeting and we came up with 3 experiments that we would do in the model, they would do in the actual work, in the actual system. And then the bad thing about this was that we said, right, for the next 3 or 6 months, we're not going to talk to each other because somehow we might let slip some idea. We want this to be an honest interaction. And why this is a bad thing is because we're very close friends. So it was not nice not being able to talk to your friend for a while. So here's the first experiment. So what we do, or what they did, was they took cells from the back and they took it away. Took those cells away. What's going to happen? Well, you could think of it, if the cells at the back are pushing the cells at the front, then if you take the cells from the back away, the front cells will not move. Alternatively, the other extreme is the cells at the front ignore the cells at the back. Okay, so just think of politicians. If you're a leader, you ignore everybody else. You just do your thing. Okay, it doesn't matter if everybody else dies. You're going where the resource is. Okay? Now, what the model predicts is something in between. The model predicts that if you remove the tissue at the back, since these cells are also in the process of taking up the chemoattractant, you change the chemoattractant behavior, and therefore you globally change the behavior of the system. In a way that we can't figure out intuitively, we have to do the model. And what the model predicts is that we will get these cells strung out like this. And that's what happens experimentally. So this is normal. Here the cells at the front are here, and in the experiment they are spread out a lot further. Okay? So the second experiment was what would happen if we took cells at the front of a donor, sorry, the other way around, if we took cells at the back of a donor and put them in the front of a host, what would happen? And here the model predicts, if you do this experiment, you put the cells here, they actually block this, and you get a slower movement than you do in real life. So here the model is wrong. And the reason why the model is wrong is we are trying to model a three-dimensional process by two-dimensional structure. Because when they do this experiment, they can't actually remove individual cells, they remove tissue. So they remove an amount of tissue, they then go to the front and they sort of push that tissue in. And when they push that tissue in, that of course doesn't totally block it, cells can move over it and around it. Okay? And that's not in the model. So the model is wrong. Yeah, but we haven't done that. What we did was a trick, a cheat, where we said, well, let's assume that it just does a little bit, it doesn't totally block, and then we find that we get the results observed experimentally. Yeah. So now the third experiment was the opposite one, which is take cells from the front and stick them in the back of the donor, of the host, and see what happens. Okay? And then, so this is where they showed, so we of course are very upset because the first experiment we were correct, the second experiment we were wrong, so we're thinking, oh dear, not very good. And then here they said, well look, when we do this experiment, this is normal, and this is the experiment, and we get some growth. And then we said, oh no, we're really sorry, because our model's wrong again. Because this is what the model predicts. Of course, you don't need to do the simulations to figure out what's going on here. When you take cells at the front, you take the tissue's got lots of VEGF, you put it in the back of the host. Now, the back of the host doesn't have VEGF, so you've put a source of VEGF. So now, instead of the cells going in this direction, the VEGF gradient is in this direction. So cells are going to stay put. So the model predicts that nothing will happen. The cells will stay where they are. So the model is wrong because this experiment here shows that the cells move. And then they said, well, actually, we should be the ones apologizing because in most of the experiments we did, nothing happened. The cells didn't move. So we thought we did the experiment wrong. And this is one of the cases where the cells did move. Okay? So you physicists will know this. I think it was Neumann or somebody who said that when a model doesn't agree with the experiment, 99 times out of 100, it's because the experiment is wrong. Okay? And say, well, if in biology, if a model doesn't agree with the experiment, maybe one time out of 100, the experiment is wrong. 99 times out of 100, the model is wrong. And here's a situation where the experiment was wrong, but the model was correct. Okay? So what we then did with this model was we asked the question, now again, if you now look at a cell at position X and radius R, and we ask the question, just in one dimension, how does the cell move? And certain probability it moves to there, certain probability it moves to there. Now, a cell has got volume, which is what particles don't have. So always in mathematical biology, we borrow from physics. And here's a situation where actually maybe borrowing from physics is not such a good idea if you want to model cell diffusing, because the cell's got volume. Particles don't have volume. And so if you now ask the question of cell moving to here will only be able to move if there's a space here. Cell moving to there can only move if there's a space there. And now if you ask the question, what is the equation that this cell density satisfies? It's this equation. So the diffusion equation would be without this term. So you're getting this extra term coming in here for low cell densities, and so you get a non-linear diffusion term. And this is making the assumption that the probability of jumping from one place to another is uniform if you make that, that the distance moved is normally distributed, so you don't automatically move a distance d, you can move some distance which is normally distributed, you get a more complicated equation. So the idea here that if you wanted to extend this model to a continuum model, you would have a nonlinear diffusion equation, and that leads to a challenge mathematically because really we don't know very much about nonlinear diffusion equations. Okay, and this is just to show simulations of the individual-based model and the continuum model and the straightforward diffusion model, and you can see that the straightforward diffusion model gets it wrong, so it is important in this situation to have this sort of term in here. Okay, so this suggests we should now be looking at nonlinear diffusion equations. And this is the references, this is the experimental work, and then that's the more mathematical description. Okay, so that was a case where this again is a continuing theme now, quite a lot of theme in mathematical biology, is if you start with an individual-based model and from there try to get the continuum model, what does the continuum model look like? Rather than say, okay, the cells seem to diffuse, we know from physics that there's a diffusion equation, we just write that down. Rather than doing it that way, start from the bottom and work up. So another thing where collective cell motion is very important is in modeling epithelial sheets. So what does that mean? Well, the epithelium is basically what covers us. And so, for example, here is a mouse embryo, and what you see here are cells, and you can see that they look this like a paving, nice sort of polygonal paving. And so people model this by using these polygons. And what they do is you can model these by putting forces at each point and solving Newton's laws of motion. So you look at this point here, F equals ma, where the forces are what's at this point, and then you move the point based on the forces, and then you recalculate. And that's called the Vertex model. There's another model which is called the Potts model, which is derived from physics ideas, which is based on deriving a Hamiltonian, and then you have cells moving to reduce an energy. And then there's a cell-centered type modeling approach. Sorry, I should just say this is the Vertex model again, where you look at the forces at any point, and then you write down the fact this is the overdamped Newton equation. And now I'll talk about the cell-centered model a little bit, which is this one here, where you consider the cells as being particles connected by springs, and then you solve Hooke's law, essentially, on how these springs move, how these centers move, and then you ask the question, well, then you do Delaunay triangulation, etc., to get these shapes again. So those are at least 3 different ways in which you can model cell sheets. So either you do Newton's law at each point here, model the forces, or you describe a Hamiltonian, and then you do a motion, and then you say, does that reduce my energy? If the answer is yes, then you accept it. If the answer is no, then the probability of accepting that motion goes down. And then the cell-centered approach, which is let's consider the cells as being point particles with springs attached and use Hooke's law. some form of Hooke's law. Okay? A natural question to ask is which one of these is correct? Okay, and the answer is that my way of doing it is correct. And your answer would be that your way of doing it is correct because your supervisor did it. And that's how it works in this field. You have schools that use one of these, and another school will use another one of these. And so you naturally might ask the question, well, why don't you compare these? Well, you can't compare them as the way I've described them because there's no common ground to compare them. Because there are certain forces and phenomena in one modeling approach that don't have their equivalent in another modeling approach. And therefore you can't do a comparison. You have to have a common framework in which to do the comparison. But in fact, that common framework turns out to be nonlinear diffusion equations. So we're back to nonlinear diffusion. Okay, and I'll show you that in a little while. Okay, so here's some work that we did where we were looking at the colon and modeling using a cell-centered model for the colon and looking at the dynamics of crypts. So there are millions of these, and they're embedded in the colon, in the face of the colon there. And what you have here is you have stem cells at the bottom, transit amplifying cells in the middle, and then differentiated cells at the top. These cells divide. These cells divide more often, and they divide about 4 times. And then as they're dividing, the cells get pushed up. And then when they reach here, and food and everything is going past, they get swept away. So in the human, there are about maybe 10 or 12 stem cells. There are a few hundred of the transit amplifying cells and a few hundred of differentiated cells. And there's, roughly speaking, 4 divisions as you move up here. Okay? And then what you can do, now people can actually track the cells, so you can see how the cells evolve. Okay? So we've been using this type of model. And again, if you think about this type of model in one dimension, you've got springs again, and you can use Newton's laws of motion. Here we are here. And then you can decide this is a linear spring or a nonlinear spring, etc., to get the forces. And you can come up with an equation for the density as a function of time and space. And these are spring constants. And what you see in the limit is you get a nonlinear diffusion equation with a very strange nonlinearity, a 1 over q squared nonlinearity. Okay. Now, while we were doing this, unknown to us, Mark Alber and his group were doing the same thing for the Potts model, for the model where you move due to a Hamiltonian. And they ended up getting the same equation, except this was not a q squared, it was a 1 minus q squared. So now for the first time, we can compare these different models. And what we can see is that if Q is normalized, Q is a normalized cell density line between 0 and 1. And what we can see is that if Q is sort of medium size, then Q and 1 minus Q, roughly speaking, the same. And it doesn't matter which model you use. But if Q is at one of the extremes, then the behavior that you get could be a model artifact. Okay? And you can understand in this system why, if Q is very small, the system breaks down. And the reason is, of course, that if Q is very small, means that the cells are very far apart. And this model system would then say that you've got 2 cells very far apart, the spring would be really stretched, and so it would really pull them in. So it would say that cells very far apart interact strongly with each other, which isn't true. So it's obvious that that this model would break down when Q tends to 0, and that's sort of exposed here by having this singular behavior at 1 over Q squared, singular behavior at Q equals 0. Okay. This is just to show that the model agrees with doing simulations, so we just leave that. And so now we have a way that if we are given cells that are attached to each other by springs, the springs can be linear or non-linear, we can tell you what the diffusion coefficient is. Likewise, if you phenomenologically, and people have done this, phenomenologically write down a nonlinear diffusion equation, we can tell you what the spring analogy is. And then, so now we can compare continuum models, the self-centered models, and the Potts models for this simple case of the cells being on a line. Okay? Now, really, things are more complicated. Raphael was talking about networks, and Roger as well. And of course, the cell properties are determined by genes. Genes code for proteins. Proteins interact in networks to give rise to the various chemicals, etc., that we observe. And so we have things like Wnt, beta-catenin, various things like this. Wnt controls the rate at which cells divide. Beta-catenin controls the physical properties of the cells. There's a gradient of Wnt up the side of the crypt. So you really need to put that in. And then you need to put these details into the model. And if you do that, and then try to do the sort of analysis that I mentioned earlier on, what you find is that you get, I don't want to go into all the details, but now you find that you get this type of model coming out where you have cell density now is a function of position, a function of time, and a function, here we're looking at beta-catenin, a function beta-catenin. And now the cell is moving through space, through time, and through beta-catenin space, because beta-catenin is not uniform. Okay, beta-catenin interacts with Wnt, and if there's a gradient of Wnt, there's going to be some sort of gradient of beta-catenin. And now you can start developing equations for how the cell density moves through this 3 space. Okay? Why is that important? So here I'm going to show you a simulation of, we want to find out, one of the questions that's posed in colorectal cancer, for example, is how can cells accumulate sufficient mutations to give you a problem? Because a cell will divide only 3 or 4 times before it leaves the crypt. So any mutation you think will get washed out. So what we've done here, so it's known that one of the earliest mutations in colorectal cancer, for about 90% of colorectal cancers, is the APC mutation. And the APC mutation affects cell adhesivity and cell proliferation. So what we've done here is we've given these cells, so these are stem cells, these are transit amplifying cells, and the stomach is up here. And we've made these cells have different adhesion and proliferation properties. And we're going to ask the question, will these cells fill all of this so they become fixed in there and so that they can get more mutations? And the answer is no, they get swept out. Okay, so if we continue to run that simulation, they would sweep away. So now what we do is we change the properties, we make them a little bit more sticky, and we make them a little bit more proliferative, and see whether this will work. Now, those of you near the front will see that the whole thing is breaking down now. these cells are extremely small, and in fact, the whole computation, if we ran this any further, would break down. Why is that? I don't know why it is, except that if we do this system here, what we find is that it turns out that for the parameter values in this system, this one predicts blow-up. So in fact, by getting this PDE approach, we can figure out in what parameter space these will have good behaviors and in what parameter space they'll have bad behaviors. Now, why is that important? Because, well, obviously we're in the computational age, and lots of people will say, well, why do mathematics anymore? Just do lots of simulations. Well, here's a situation where we have lots of parameters, and you'd have to literally do billions of simulations in order to get some idea of how this behaves in parameter space. And when you get the sort of behavior that I mentioned here, you have no idea why that behavior was like that. The PDE tells you precisely why the behavior is like that, and it tells you the parameter regime in which you'll get that behavior. So it's very powerful if you can do it. Okay? So here's an example here where here's a cell, and we're going to follow its progeny. And look what happens as we go along. It looks like it's going to take over. Oops, but then it doesn't. And here's another one. Doesn't look much different to this, but it does take over. And this is called monoclonal conversion. It's the idea that these crypts are all of one clone. And so what we can do then is we can find out, using this type of model, is how long does it take for there to be monoclonal conversion? And here we can find out here that all the crypts are monoclonal. Now there's a lot of controversy about stem cells and what this model, so there's some people who say that stem cells are immortal. So for example, they're saying that this cell here stays there all the time. And there are other people who say there's no such thing. And what this model says is that there's no such thing. That stem cell that began everything gets washed out. This tells you the time it takes to get washed out. Time and days to which they all get washed out. But it gets replaced by another stem cell. Okay. And then you can ask questions. What's, if you've got the proliferation rate, what's the probability of fixing so that everything becomes that cell type, so that mutant overtakes everything? And here, if you've got adhesion, what's the probability that the mutant adhesion, as you change the adhesion, what's the probability you'll take over? And one thing I should point out here, this is, as the cells divide and they move up the crypt, at some point they stop dividing. And this is telling us how does the probability of the mutant taking over the crypt depend on the height at which the cell stops dividing. And what you can see here is that this is a very spatial phenomenon. And models have been proposed for this which are purely temporal. And this is telling us that those models probably are a good first pass, but but don't take into account everything. Space is important as well. Okay, and you can see that here you ask the question, well, if the cell stops dividing when it reaches this point, then what's the probability it'll be fixed, versus if it stops dividing at this point, what's the probability it'll be fixed? You can ask questions like this, which I won't go into the details of that. Okay, and then within this work, my colleagues, mainly in computational science, have devised this open source modeling framework that models individual cell-based models, so you can use whichever model you want. POTS, the Cellcentered model, the Vertex model, and you can put any ODE you want to put inside those cells, and then you can see what happens. And it's modular, so you can change things back and forth. And this is freely available. It's called CHASTE for cancer, heart, and soft tissue environment, because it's used by the heart modelers as well. So for the last minute or so, I just want to mention something about something I talked about earlier on, about the idea that if you have a sort of mean field approach, which sort of assumes that you don't have volume, what happens when you correct that and do it properly? And this is work very recently we've been doing, and because of time I don't want to go into the details. What you do then is you say, well, okay, let's now consider lattice, regular lattice, square lattice, and we ask the question, what's the probability density that lattice L has got species I in it? Well, it's the probability that lattice L had nothing in it, but its neighbor had one cell in it, and that one moved to there, minus the probability that the species was present in it, but it moved to the neighbor because the neighbor was free, plus the probability that the neighbor divided, moved into here, minus the probability that the cell died. So you see here, if we want to work out the probability density of one lattice site, we have to look at pairs of lattice sites. So we need to write down equations for pairs of lattice sites, and as usual, this leads to triplets. So then we have to have some sort of moment closure going on, okay? And then we can also do a mean field. The mean field of that would be this one, okay? So if we consider two species interacting with each other, the mean field would be two equations that look like this, which you can solve analytically almost, okay? The correct version would consist of two ODEs of this form, plus 3 PDEs describing the correlations. So it's much more complicated. So what's the point of doing that? Well, the point is that this gives you the wrong answer and this gives you the right answer. Okay? And here's an example. I'll just take one of them. This is for certain parameter values, and this is the individual-based model, and two species, A and B, and the prediction is over time you get this. This is the mean field approximation, and this is what I just described, the correlation. Now you can look at that and you can say, well, there's not much difference there. You know, biology wouldn't be able to pick that difference out, so you know, what's the point of doing all that extra work? But here's a case where when you do the actual simulation of the system, you get that A wins and species B goes extinct. Doing the way I just described, A wins, species B goes extinct, using the mean field, B wins and A goes extinct. So the mean field gets it totally wrong. And that's a real pain in the bum, because that means that we have to now worry about solving these very complicated equations instead of solving the very simple equations. Okay, so what I've tried to do there is to illustrate with the first 3 examples how you end up getting different approaches, like a PDE approach versus a hybrid cell-based approach versus a totally cell-based approach, and that the underlying theme in all of that is a non-linear diffusion equation. where the non-linearity in the diffusion really picks out what you're saying about what's happening at the microscopic level. And then this last piece of work, where showing that the mean field and doing it properly in inverted commas, you can get completely different results. Now, the problem with the mean field approach, the way we've used it up until now, is that it can only work if you've got symmetric initial conditions. So some of the properties that we use to get those equations assume certain symmetries, and those symmetries aren't present, for example, in the initial conditions in the first system that we looked at. So it's why we can't apply this to the first system. So one of the challenges now is to try and take this sort of corrected mean field approach and try to generalize it so that it can be applied in more areas. So I'll stop there and thank you for your time.